And I think it'll be exciting because resiliency is a hot topic, especially pun intended with climate change. Things are changing. And so this is the whole point of resilience is how do systems respond to change? I want to get us warmed up. Think about like, what is your definition of resilience? What is resilience and why is it important? You can either unmute yourself and speak out, drop something in the chat, and extra points if you have a story or an example that you want to share. So I'll give you a couple minutes to get your wheels turning. And when you think about resilience, what does that mean? Okay. We've got example of a crop recovering from heat, yeah, ability to bounce back, to respond to stress. A buzzword that helps make us feel good. Okay, seeing a lot of withstand stress, bounce back. Specific example, crops being able to recover after a major temperature stress. Oh, self-renew, that's a very good word. Oh, adaptability to shock and stress. So I'm seeing withstand, adaptability, bounce back. Okay. Anyone have an example, like a personal story that they've seen on their farm? Okay, well, we can get back to that later. So, ooh, weeds, super interesting. The weeds are the resilient ones, or are they the stress? Anyhow, so seeing some commonalities. So a major way that it's talked about in the ecological literature is as stability. So I like the phrase the tendency of a community, and this comes from ecology. So a lot of the literature is about community membership or composition, but to return to a stable state. So visualized as these kind of terrain models, if you picture a ball in a basin, like a little shock will move it up a little bit and it'll move right back. A bigger shock will move it a bit more out of its stable state and then it will move right back. If you get a really big shock, it may kind of move it over the hill and into another basin. So an example of this might be like you have a rainforest and it has a couple of drought years. There's some plants that die, but their seeds are in the seed bank. The major conditions haven't changed. And so some functions will change, but eventually it'll tend to return to that stable state versus like a multi-year drought or like the rainforest is bulldozed and they plant soybeans. The major environment has now changed. And so this system cannot return to where it was before and there's a new stable state. So this is a common way to think about it in ecology, and it has two components that you guys brought up in your definitions. First of all, resistance. So how does it resist the change? How far does it move when it's disturbed? And then resilience is the second part. So it's been disturbed. How quickly does it bounce back? How completely does it go back to its normal state? So we'll be talking about these a lot. So when I say how far it moves, what is it? There's a lot of different functions that people measure. So a big one is community composition, whether that's like species in a grassland or microbial community in a soil. The diversity. Also, people talk about it in terms of some community function. For example, nitrogen mineralization in a soil system can be changed by a stress, and then you can measure how it goes back to its normal state. And then kind of on a higher level, some broader symptom function, usually something that you want to happen. So something like crop yield or the ability of landscape or a soil to store and provide water, something like that. And there are others, but these are just examples of things that people measure, how far they move, how they get back. So here's a local example. This is from Russell Ranch, our sadly now no longer functioning long-term research station. And this group of researchers just measured the yields over time in a corn tomato system that there was a conventional system, organic that got manure and a cover crop, and then a kind of low input system that also had a cover crop. And so when they analyzed the yields over time, if you see those triangles, that's the organic system, they had a higher stability than either of the other two. And so they didn't have the high highs or the low lows. So less fluctuation and then also higher resistance. You'll notice there's a couple of years where both of the other systems had really bad years. Those were stressful conditions that didn't affect the organic as much. And so these were considered to have a high yield resilience. But resilience is very complicated. And this is a theme that will keep coming up because remember, these were corn tomato rotations. So every year, corn tomato on the same plot, same soil. But when it was corn that was growing, again, these triangles are the organic system. It actually had a lower resilience. It had more fluctuations and more potential for crop failure. So just to say that like resilience isn't this kind of magical property that's there on its own, which we sort of tend to think about it as, but it's saying like this particular organism, how well does it do this thing that we want it to do in this particular environmental condition? So in this case, the same environmental conditions were better at growing tomato than they were at growing corn. The risks were different, the challenges were different, so the stability was different. So I did a postdoc in Tennessee where I got the opportunity to look at the links between soil management, soil health properties, and climate resilience. So the basic idea was choose 3 very different management systems on the same soil type, measure a ton of soil health parameters, measure resilience to heat and drought stress under controlled conditions, and then see if resilience relates to soil health and which aspects of soil health does it relate to. So I used a fairly common metric. There's a lot of ways to measure resilience. This is just one of them. But the idea is that you have a sample. I used undisturbed soil cores. And you keep some of them at pretty optimum conditions, reasonable temperature and moisture that reflect fairly normal conditions for that soil. And you keep that as the unstressed control. And then you subject other samples to stress over time. So you measure some kind of function, for example, let's say soil respiration. At the maximum stress. So how different the stress is from the control. At the maximum stress, we call that resistance. How far does it go when you poke it? And then you take away the stress. You bring it back to unstressed conditions, and then you measure your function over time to see how quickly does it start to look like the control again. So this has its issues. It's one way of measuring resilience. This was my experimental design. So I had a conventional corn soybean system. It actually been no-till for a long time because that's what's common in Tennessee, but otherwise synthetic inputs, once in a while a cover crop. I had an organic system that was run for many decades on the Rodale system. So a 6-year rotation, corn, soybean, and 4 years of pasture. The pasture was grazed before the corn. It was tilled in, get manure addition, and so yeah, cover crops in all of the non-pasture phases. So really observing all the soil health principles for several decades. And then it wasn't historically no-till, but in a new owner had taken over and he had started to try. So this was his first year of trying no-till. And then kind of around the organic system, there was a strip of secondary forest. So I also took samples from there. So measured a lot of different biological and physical and chemical properties. So I'm measuring microbial communities, enzymes, soil carbon, doing little tests of how well they mineralize, stuff that I give them. And then also physical properties like porosity. And I was especially interested in thermal properties. So how well do they store heat? How does heat spread? And then also chemical properties, NPK, pH, and so on. So with these, I run a principal components analysis, which is basically just a statistical test that identifies what are underlying factors that differentiate the systems. And then I have these undisturbed soil cores that I either leave at a nice soil temperature, nice soil moisture in an incubator, or I let them slowly dry out in an incubator over time, so subjecting them to drought stress. And then other ones, I subject them to drought stress, and then I move them into a hot incubator. So these have both drought and heat. I measure a lot of different measurements of carbon and nitrogen cycling, both at the maximum stress, and then I bring them up to a good soil moisture and store them at the same temperature as the controls for many weeks, and measure the resilience twice, and the same indice or the same properties. So these are things like enzymes, microbial biomass, respiration, nitrogen mineralization, and so on. So then I have this data for both the control and the stressed samples, and I just use these to calculate an index. So just average them together. How different on average were these cycling functions with the treatment and the control? And then, these give me an index of carbon and nitrogen cycling. And then I just correlated them against these factors that are differentiating the soils. So it'll become clearer later when I talk about the results. But first of all, I want to go into a couple of hypotheses. Like, are healthy soils more resilient? We always kind of assume that they are. And there's one hypothesis that says yes, because they depend on the capacity of a system to internally regulate itself. And so the more internal regulation capacity there is, the more resistant and resilient it will be to stress. So an example of this might be if a soil has good soil, a lot of soil organic matter, it has good structure, good water holding capacity. And so then because it has these good internal regulation capacity for storing water, if there's a drought, then this system will be less stressed than one that's not able to hold water so well. So the stress will be less because it has this good capacity to regulate itself. But wait, there's a totally opposite hypothesis that says, all right, so you have these microbes, they have a good home, they have a lot of food, they're well taken care of, they're from a healthy soil. And then suddenly you subject them to conditions that they've never been under, where they don't have shelter, they don't have food, they don't have oxygen or whatever. Like, who's going to do better? The one that's not used to any stress, the one who just they don't have the community members there that are able and ready to function under that stress, versus a community that sees that stress all the time. They have the tools, they are able to deal with it. So this hypothesis says that microbes from non-stressful environments probably have much worse resilience or resistance. So they'll move more when you poke them, but they might have good resilience. And that's because if the good conditions go back to being good conditions, that will provide a better environment for regrowth and for rebuilding the community function than in a more starved environment or more hazardous environment. And so that's kind of similar to hypothesis number 3. I'm sorry, this is kind of a busy slide. But basically, if you look at the y-axis here, it's saying that resistance and resilience, it's a trade-off. You either have high resistance or high resilience, but you don't have both. And in this hypothesis, it says it depends a lot on your initial community. So kind of tortoise or the hare. If you have a lot of organisms that are kind of live fast, die young, they have simpler genetics, they're fast growing, they're able to like respond quickly to resource availability. These will suck at dealing with stress, so they'll have low resistance, but they'll be really good at bouncing back. So they'll have high resilience versus on the tortoise side, if you have the slow but steady wins the race, you have more complex genetics, they're investing more into their stress responses, so they're slower growing. They'll be able to withstand a stress better, but they won't be as good at growing back. And then it's also mediated by the nutrients that are available. So like if a microbial community needs to grow back, it needs things like carbon and phosphorus and things like this. And so the more nutrient rich that an environment is, the better resilience will be. So with those 3 hypotheses in mind, let's go forward with the experiment. So my conventional soil, this is what it looked like. Notice there's some soil cracking in there. Notice the thin residue cover, redder color. Organic, it had a thick residue cover. It was a very spongy soil. It had higher moisture. You could tell just by feeling it, even though the fields were very close to each other. In my forest soil, this was also very spongy soil, very thick leaf cover, many roots, lots of soil fauna. So very different soils, and that's what showed up when I did my 10 million soil health tests. So I basically characterize each of them here. The forest soil was not an ag soil. It had the highest porosity, and like I was predicting, it had that went with it had a high heat capacity and resistivity, so heat didn't travel far in this soil. Aggregation wasn't so good. It was also a lighter textured, but also just the organic matter wasn't as decomposed. So it had a lot of relatively less decomposed organic matter, and actually not as much organic matter as the organic system, and less microbial activity, less microbial biomass. I wasn't able to measure bacterial to fungal ratios, but it did have a higher fungal diversity and also had a lot of Basidiomycetes, so kind of forest mushrooms, kind of fungi. And it was a very acidic soil. This is Tennessee, high precipitation, it's not being limed, and really low fertility. So things like P and K. So some like healthy soil characteristics from an ag perspective, some unhealthy soil characteristics, it's not an ag soil. And so when I separated out my 3 systems in this graph on the right, the forest is the dark blue, and the biggest difference between systems was between ag and non-ag. So I'm calling this arableness that separates out the forest. My organic soil was like a classically healthy soil, very high fertility, very high soil organic matter, lots of enzyme activity, lots of microbes. The total available N was pretty low, but it also had a standing corn crop, so probably that tightly coupled nitrogen and carbon cycling, very good aggregation, highest water holding capacity. This was a classically healthy soil. The conventional I'd characterize as fragile, had high chemical fertility and high pH. Both of the ag systems are being limed, I should mention. And especially high nitrate, which this also has a standing corn crop. So that means they're probably over fertilizing, very low soil organic matter, and very low biological activity, poor soil structure. So I'd call this a fragile soil. So how did this show up when I measured resistance and resilience? Unsurprisingly, it's complicated. A couple of things that I want to point out. So these graphs here, this is again my kind of bundle of all my carbon and nitrogen cycling functions, how far they moved when they were poked, that's resistance, how they started to resemble the control again, that's resilience. And I found that resistance depended more on treatment than on site. So when we added heat, none of these soil health properties mattered. They all suffered when we added heat, but some more than others. When we didn't add heat, for drought only, there was a tendency to decrease. So forest was the highest, then organic, then conventional. And so there was a pretty strong correlation to my metric for arableness because the forest tended to have the highest resistance. Resilience was the opposite. It differed more by site than by treatment. So resilience is really where our soil health, our different soil health characteristics came into play. And it didn't actually matter a ton for treatment, except for a couple of examples that I'll go into. And this was true for both carbon and nitrogen cycling and resilience. I don't show the data, but pH was a really strong factor here. Notice that the resilience in the forest soil is basically zero. And this correlated strongly to pH and some of the other nutrients like phosphorus, and then also just kind of general soil health. So overall, the organic system tended to be the most resilient, not always significantly, but that was the general tendency. Nitrogen cycling was interesting. It was always less resilient than carbon cycling. And if you look at resistance in the forest soil, when you just added drought, it was far and away the most resistant. When you added drought plus heat, it was far and away the least resistant. So heat really exacerbated the effect of drought on the resistance of the forest system. If you look at conventional nitrogen cycling resilience, so how it recovered, this negative value really means that in the drought-stressed conventional system, it actually looked worse after the recovery period than it did at maximum stress. So it did not recover at all. Heat decreased that effect. And I'll talk a little bit about that later. So how did this feed into our hypotheses? We have our first warm and fuzzy hypothesis, internal regulation, more resilience. And I think we saw that in our conventional system nitrogen cycling. This is, again, very low organic matter, low biological activity, very poor nitrogen cycling ability. It's getting all of its nitrogen synthetically. And we saw no recovery of the nitrogen cycle after the drought stress. And I think that's because basically there's no carbon there to soak up that nitrogen. There's no carbon to really fund the growth, fund the regrowth and regeneration of that microbial community, except in this short term, when you add heat, then what we noticed is that there was a big bloom of available carbon when we added heat. And that's because we popped all the microbes. They spilled all their guts out. And so these poor carbon-starved microbes in this conventional system, they have a lot of extra carbon when we added the heat. And so I think that's why in this case, we were able to see better resilience under heat stress. And then also we notice that when there's like when a soil gets really dry and then you add water, it tends to bring soil organic matter that's trapped on soil surfaces off of it. So that's some extra soil carbon. But I'm sure that if we looked at this over time, if we did this experiment many times, it wouldn't be that we're looking at higher heat gives a higher resilience. We'd be looking at the slow depletion of what little capacity for bounce back that we have. So scale really matters. And I'm going to be coming back to that over and over again. Scale matters. What about hypothesis number 2, that we'd see low resistance in soils unused to stress? I think this is a good explanation for our forest soil nitrogen cycling. We saw very good resistance to drought, but very poor when heat was also added, which if you think about it, drought is a very common condition in this forest soil, but heat is not. It's permanently shaded. It always has a residue cover. So these poor soils where I took off all their residue and I put them in the blazing sun were exposed to conditions that they would never see in nature unless something really drastic happened. And so this is really a false experiment because it's saying what would happen if I took them out of this system where they're meant to do well. And so that's why I said, are healthy soils resilient is a trick question, because what I should be asking is, are healthy systems resilient? Are we keeping the protections there that would allow a functioning soil to remain, in this case, resistant? Also, it was interesting that it happened, especially with the nitrogen cycle, because these are soils that I think, based on various indicators, even though I didn't measure it, are probably going to be more fungally dominated. And fungi are really good at dealing with drought because their little hyphae are able to bridge across soil gaps. They're more competitive under drought, but heat really screws them up. And they're also really important in nitrogen cycling. So this is my theory about what's happening in the forest system. Hypothesis number 3, not positive about. I'm not super convinced. We did see a tendency in the conventional system it always had the lowest resistance. Its resilience was okay, except nitrogen to drought, and it has a good pH, rich in mineral nutrients, but poor soil organic matter. So kind of forest soil had the high resistance, low resilience, nutrient poor. So its general response kind of fits with this model. I didn't see, when I looked at the microbial communities, I didn't see any evidence that they were more dominated by the tortoise or the hare. So that part of this hypothesis I didn't see. But organic system, that was interesting because it had pretty good resistance and it had pretty good resilience. And so I think of it as kind of the Goldilocks position. If it is a trade-off, it's able to be in the middle. It doesn't have, it never had a big crash for either one, for any of the nutrients. And so I would say in that case that this healthy soil that had both the good agricultural properties, high pH, nutrients, high soil organic matter, was able to support, and also probably had less year-round soil cover than the forest soil as well, but was able to be more resilient. But there's a sad part of this story, which is the organic system this crop could not be harvested. The grower who was experimenting with no-till, terrible problems with insects, the nitrogen wasn't enough, so the yields were not good. So again, thinking about resistance, resilience, it really matters. What scale are you looking at? What are you measuring? And basically, why do you care? So all of these observations, I'm kind of exploring towards a hypothesis for, based on my favorite metaphor in agriculture, which is the leaky bucket, if you're familiar with this. So this was developed in the soil fertility world and says that yield is going to be limited by whatever nutrient that it needs that is in that it doesn't have enough of. So for example, you can have a soil with a lot of potassium, but if it doesn't have enough nitrogen, you won't get a good yield because that's going to be limiting. And I think that this can be applied to a lot of different functions. So I want to say that whatever function you want the soil to do depends on the microbial community that's present or the broader soil community and their ability to perform that function. So limitations will depend on the genetic potential in that community. For example, like if you put the community underwater, is there anybody there who is able to perform under anaerobic conditions? That kind of thing. So it will depend on the genetic potential in that community to perform whatever function you're looking at under present environmental conditions, whether that's their normal conditions, their conditions under stress, their conditions after you remove the stress. Are the guys who are there able to do the thing that you're looking at? And that'll depend on who is there initially, what function you're looking at, the type of stress. Is it something that that community regularly sees, the environment? So that goes in with hypothesis 2 and 3. But then there's a lot of limited, like kind of commonly limiting factors, things that most microbes need, and a lot of them have kind of similar needs. And so soils with good values of many of these common limiting factors, so things like soil organic carbon or pH or fertility or water holding, or good water holding properties, aeration, will tend to function best under a wider range of stressful conditions. And then that kind of one step up, are we considering this soil as part of a broader system? Conditions that buffer the community from sudden changes or extremes will promote stability. So things like good soil cover or something that aren't necessarily part of the soil. So I wanted to end by just emphasizing resilience as a matter of scale. And so thinking about it first from the perspective of an individual microbe. What does that microbe need in order to do what it does best, or in order to function? So it's oxygen, it's some kind of like a energy source, some kind of a shelter from harsh environmental conditions. What protects them when there's a change that's hard for them? And then what resources do they need to recover? And moving up from an individual microbe, local populations of microbes, they'll have some of the same needs, and some of the same protections, but some will be different. So for example, like having a diverse group, so some are able to respond to the stress when it comes. Well, maybe they need a diversity of carbon sources or a diversity of microenvironments, and so on, going up the stage. So taking into account the whole soil community, the soil food web, and then up further into the agroecosystem. So including the crops, the insects, the weeds, the water, and so on, and then up and up and up. So that's what I've got in terms of presentation. I'm happy to either just do free-form questions, or I also have a discussion quest, like a broad discussion question if people want to do that. And so the discussion question is, thinking back to when you were talking about what resilience is to you, why is it important? If we want to design and test resilient farming systems, what scale should we be looking at? We're saying our stress is a changing climate. What scale is going to be the most important? Maybe multiple scales are. And what functions should we be looking at? What are the limiting factors? What are the buffers or protections? So yeah, either we could, that's an idea for discussion, or we can also just general comments or questions.
Wonderful. Thank you, Patricia.
Thanks
Thank you
very
for
much.
helping us.